Hello and good afternoon. I can see Anusha and Reena already live for the session, and I'm sure the others are going to join very soon. So how are you doing, Anusha and Reena? How was your Sunday? And also let me know if this audio is clear. Can you hear me clearly? We'll continue with our chapter, that is breathing and exchange of gases. Okay, Reena, thank you. And we'll have a quick recap of what we did earlier, and then we'll start with oxygen dissociation curve, then Bohr's effect, control of respiration, and we will also start the disorders associated with the respiratory system, because this is the last part of this chapter. And then after we finish this chapter, then we'll have a quiz. Okay, I can see a new name, Zara. Hello, Zara. Are you a new student to this class? Are you attending this class for the first time, Zara? Okay. And why did you start so late? Because I think today and by tomorrow, a little part, we will finish this chapter. We have already done some chapters earlier. Oh, you watched the previous lectures, but those were probably the recorded ones, right? Okay. Good afternoon, Anju. So, how was your weekend? How was the Sunday? Was it like a routine day, or you did something special? I think it's Diwali time, but that's why students are becoming little lazy. They're joining in little late. Routine day plus playing. Which sport do you play, Anju? Do you play some sport, or is it like just the normal thing? Oh, Anju, you play cricket. Very good. So, are you representing some like the city or the state or something? Good afternoon, Irene. How are you? Oh, just your family. Okay, all right. But I'm sure that must have been fun, relaxing also. Okay, Helen, that's good. All right, so let us get started. And what we have planned for today is a recap of what we did, plus doubt solving in case if you have any. Then oxygen dissociation curve, we'll talk about Bohr's effect. control of respiration, chemical control and nervous control, and then if time permits, maybe we'll start with the disorders associated with respiratory system. Okay, so let us get started with the basic things that we have to talk of. The channel where we are right now is Let's Crack NEET UG English. And those who are not aware of their bio teacher, that is me. So this is some information about your bio teacher and the platform that we are using. This is the app that you need to install because very soon we will be starting a plus program, and those will be available only on this app. So you can just go to your app store and get this app installed. So it's very simple, and I'm sure you're all aware of it, and some of you probably have already installed, because I can see some students who are coming on my special classes as well as here. And this is the plus subscription that we normally talk of. It comes with some very nice features, like you have daily live classes, plus you can watch all the recorded ones as some of you have done. Then you also get unlimited access to the lectures of other teachers. So it is not only just one thing, one tutor, you can watch the videos of other teachers. There are quizzes, live tests conducted, and then here there is a structured course. Like when I start with plus, maybe it's going to be just a 3-month recap or 3-month crash course or something. So it is going to be all structured, like on which day what will be done, when we are going to have the quizzes and all those things. And this is a newly added subscription that is called Iconic. It has some added features like personal coach and test analysis, and this will give you a comparative thing. So Iconic has every feature of plus and the added things. These are the 4 added things which you get in the Iconic one. And this is just to give you a comparative subscription plan. Like in case of plus, you have the option of picking a short term, like a monthly subscription. You can go for 3, 6 or 12 month or even 24. And the longer the duration is, better the off or discount is. Like here you get only 17% off. You go for 6 months, you get 33%. If you go for this, you get 54%, and here it is 67%. And don't forget to use this code so that you are eligible for 10% discount. And same is the case with Iconic. So if you go for a little longer subscription, then there is 14%, and then if it is 24 months, that is 2 years, it is 34%. And again here also you can use the same code to get the 10% discount. Okay, let us come to the doubts which you can ask now, and those are from this chapter of breathing and exchange of gases, everything. Because as soon as we finish the chapter, we will have a quiz or a test, whatever you call it. So is there anything which is not clear from the structure of respiration, respiratory surface, structure of respiratory system, nasal chamber, trachea, bronchi, everything? You can ask doubt from anything from this chapter. Hiren says there is no doubt. I think everything is absolutely clear, Hiren, that means. And what about others, those who have joined in? Reena, Zara, Anju? No doubt, Hiren says everything is absolutely clear. Perfect. Zara has a question, ma'am. Did all the 4 oxygen molecules enter the tissues or only 2? See, oxygen, when we talk about oxygen dissociation curve, all the oxygen molecules do not enter the tissue. Some oxygen remains attached with hemoglobin, and that is why deoxygenated blood also has oxygen in it. That will be clear after we talk about the oxygen dissociation curve. Okay, so Anju says no queries, and Zara, your question will be answered after this oxygen dissociation curve is discussed. So that's good that you don't have any doubt or any query. So we can get started with oxygen dissociation curve. See, this is how the oxygen dissociation curve looks. This image has been taken from your NCERT, and I'm sure you all have seen this image. Now, who has a doubt related to this oxygen dissociation curve? Such shape is called a sigmoid curve. When the graph is of this shape, it is called sigmoid. It is like this. So oxygen dissociation curve is sigmoid. And when you say it is going like this, here, you say it is moving towards right. The graph is moving towards right. Himanshu, hello Himanshu. So do you know what exactly is this oxygen dissociation curve? When we talk of oxygen dissociation, we actually talk about dissociation of oxyhemoglobin. Sigmoid is S-shaped. There are two shapes of graphs. One is like it starts like this and goes up, continuously increases. And the other is starts like this and then it becomes a steady one. So this is called sigmoid and this is called J-shaped. So normally all the processes, everything is sigmoid, because at some stage the process becomes more or less steady. It is going to go increasing, but after some time it will become steady. Okay, now I'm going to show you something else, and I want to know whether you have read this or not. This is from our NCERT. This whole thing I have just taken from our NCERT as it is. Now here the information which is given is a mixed information about hemoglobin also, about oxygen, hemoglobin binding also, and the dissociation also. So let us go over this, understand this, and then we will see the interpretation of the same in the form of that graph. Now hemoglobin is a red colored iron containing pigment, and it is present in RBC. We all know this. Okay? Oxygen can bind with hemoglobin in a reversible manner to form oxyhemoglobin. What does this mean? It means that equation which we wrote, oxygen plus hemoglobin, we made the reversible arrow, that is oxyhemoglobin. That means it is a reversible reaction. Okay? Now, the next point is again what we have already talked of, and that is each hemoglobin molecule can carry a maximum of 4 molecules of oxygen. So we made this hemoglobin, we wrote O2, O2, O2 and O2. And this is oxyhemoglobin. So one molecule of hemoglobin can bind with 4 oxygen molecules. Now comes the next and another very, very important part. And that is binding of oxygen with hemoglobin is primarily related to partial pressure of oxygen. So when partial pressure of oxygen is more, more and more oxygen is going to bind with hemoglobin. Okay? Now, partial pressure of carbon dioxide, hydrogen ions or hydrogen ion concentration and temperature are other factors which can interfere with this binding. So there are 4 things which are responsible for binding of oxygen to hemoglobin. Oxygen binding to hemoglobin. Can we say factors responsible for formation of oxyhemoglobin? Because if oxygen binds with hemoglobin, that complex is called oxyhemoglobin. So for formation of oxyhemoglobin, what are the conditions? Partial pressure of oxygen is a factor. Partial pressure of carbon dioxide is also a factor. H+ ion concentration is a factor, and one more, that is temperature. Now more temperature, less temperature, more H+, less, that we will see. Okay? Now the next important thing is sigmoid curve is obtained when percentage saturation of hemoglobin with oxygen is plotted against PO2, and this is the graph that we were talking about. So if you plot a graph and that is percentage saturation of hemoglobin with oxygen partial pressure, if you plot this, then that graph is going to be a sigmoid graph or sigmoid curve. This curve is called oxygen dissociation curve. Okay, let us go back to this curve, and this is the same oxygen dissociation curve that we are talking about. It is percentage saturation of hemoglobin with oxygen, and this is the partial pressure of oxygen. So here we are talking of PO2 and here we are talking of percent saturation. Let us take few figures. If partial pressure of oxygen is 20, PO2 is 20, then how much hemoglobin is attached with oxygen? That is what is saturation. Saturation means if there is, say, 100 molecules of hemoglobin, that what percentage of hemoglobin are attached or bound with oxygen. So here if you plot this, you will find that it comes to some 30. So percent saturation is 30. That means 30% hemoglobin is still attached with oxygen. Now let us talk about 40. So if PO2 is 40, then somewhere here, that is around 70, this will be 70 here, so somewhere around 74, 75, so it is approximately 74% oxyhemoglobin is still there. Where do we find this 40% partial pressure of oxygen? Don't you think this partial pressure of oxygen of 40 is in the tissue? Tissue has used up the oxygen and there is more and more of carbon dioxide. 45 was the PCO2 and 40 is PO2. So when the partial pressure of oxygen is less, still oxyhemoglobin, saturation of hemoglobin, that means 74% is still oxygenated. Let us come to 60, it is about 90. Now, why we have taken these figures, that is partial pressures? Let us take one more, 90%, because if you remember, alveolar air has partial pressure of 104 and the oxygenated blood has 95, and that is why we are talking about this. So now if PO2 is about 95, which is at the lungs, it is almost 100% saturation. So this is the blood which we call oxygenated blood, right? Let me just draw a diagram for you, which we have already talked of. We have seen this diagram. Here is this alveolus and here are the tissues. Now when oxygen diffuses in, PO2 is 95, right? And here we say the blood is oxygenated blood. Here at the tissue, PO2 is 40 and oxygen diffuses into the tissue, and the blood which leaves the tissue is having PO2 40, and this is called deoxygenated. Now oxygenated means hemoglobin has oxygen. Deoxygenated means hemoglobin is not having oxygen, but that is not the situation. When we say PO2 is 40 and the blood is supposedly deoxygenated, that time also hemoglobin saturation is about 70% plus. So though we call that deoxygenated, but it is never that complete oxygen is going to diffuse. And that was your question, Zara, that at or if deoxygenated blood is there, or if at tissue oxygen is diffusing from the blood into the tissue, complete oxygen is never going to diffuse. That means hemoglobin will still remain attached with 1 or 2 oxygen molecules. Okay, let us come back to this paragraph. Now, it says this oxygen dissociation curve is helpful in the studying of effect of factors like PCO2, H ion concentration, etc. on binding of oxygen with hemoglobin. Now there is a question which is invariably asked. What are those conditions which are essential or helpful in formation of oxyhemoglobin, and which are those conditions which are helpful in dissociation of oxyhemoglobin? And why this dissociation is so important? Because if dissociation takes place, then only it is oxyhemoglobin which is coming here. So if dissociation takes place, then hemoglobin will be set free and oxygen will be released into the tissue. That is why this dissociation is very, very, very important. Now, what are the factors which are responsible for dissociation, and where is this dissociation taking place? Dissociation of oxyhemoglobin takes place at the tissue level. Now let us see what are the conditions here. PO2 is low. See, at the tissue PO2 is low, 1. At the tissues carbon dioxide is more, so PCO2 was 45, it can be 48 also depending upon the activity of the tissue. So here PCO2 is high. These are the conditions when oxygen is going to be released or oxyhemoglobin is going to dissociate. When is that going to happen? PO2 is low, PCO2 is high. Temperature. What about temperature? Here we breathe in air. So it is the air from the atmosphere which is coming in. Though our our respiratory chamber in the nasal chamber brings it closer to the body temperature, but still if it is coming from outside, here it is fresh air. And what is happening at the tissue? There are various metabolic reactions which are taking place, and in these reactions heat is generated. So comparatively at the tissue level, temperature is going to be slightly higher. So higher temperature, it's not like very high, but if you're comparing at lung and the tissue level, then at the tissue level the temperature is definitely going to be higher. Now the last thing, H ion concentration. Can you tell me what is happening here? H ion should be more or less? For this, what I'll do is I'll leave this here and then we talk about another process. Here is an RBC which has reached. So this deoxygenated blood comes here and this is the RBC. And here is plasma. This is happening closer to the lung. What is in the plasma? HCO3, bicarbonate ions, right? So now if you want oxygen to be exhaled, then these bicarbonate ions will move inside the RBC. You remember what happened in chloride shift? That means here, if we are talking about chloride shift, where is chloride shift taking place? Here. So let us draw one RBC here also. What happened? Carbon dioxide diffused in, it reacted with water to form carbonic acid. This carbonic acid dissociates into H plus and HCO3. HCO3 come out, and to counter their number, chloride ions move in. So we called it chloride shift. Some H ions are also there and these H ions will also be released here. So here H ions will increase. Okay, let us take this. This bicarbonate goes in, the H also goes in, and this results into formation of H2CO3. H2CO3 dissociates in presence of enzyme which is again in RBC, and that is carbonic anhydrase into water and carbon dioxide, and these two are exhaled. So if bicarbonate is going in, the chloride ions which were inside, they should be pumped out, so chloride ions will come out. Here you called it chloride shift. Here also there is chloride moving, so you call it positive chloride shift, and this is known as the negative chloride shift. The positive is known as hamburger's shift, and this is called only negative chloride shift. So hamburger shift or positive are same, and chloride shift is only symbol. So now if H ions from here are going into the RBC, what is happening to the H ion concentration here? H ion concentration decreases. Here H ions will also come out, so H ion concentration increases. So dissociation, what are the four factors which would help in dissociation of oxyhemoglobin, which takes place at tissue? Low partial pressure of oxygen, high PCO2, higher temperature and higher H plus. Now this is dissociation. So now if you're talking about factors for formation of oxyhemoglobin, what are those factors? First is low H ion concentration. lower temperature, PCO2 low, PO2 high. So just reverse. You don't have to mug it up. You have to just understand that this is happening here at the tissue level. And what are those conditions which prevail at tissue level? Those are the conditions required for dissociation of oxyhemoglobin. Is it clear that what exactly is the impact of these conditions on dissociation of oxyhemoglobin, and why should it dissociate? Because unless and until oxyhemoglobin dissociates, oxygen will not be released and it will not be available to the tissues. Plus, you also want this hemoglobin to be set free so that it can bind with carbon dioxide, because now it is going to take carbon dioxide. So is oxygen dissociation curve clear? Is negative chloride shift clear? So normal chloride shift, if it is written, we are talking about this one. If only chloride shift is written. If positive is written, we are still talking about the same. If Hamburger's effect is written, we are still talking about the same. But what happens at the lung level to release carbon dioxide is reverse of this. So you call it negative chloride shift. So normal chloride shift or positive takes place at the tissue level and negative takes place at the lung level. One review on oxygen dissociation curve. This is what is the, not the question, this is what Hiren says, that he wants one review. Okay, Anju is saying negative chloride shift. All right. Anything else? Amisha, everything clear? Okay, let me do one thing. Let us do this oxygen dissociation curve one more time. Everything fresh. So we will understand the dissociation, the factors, as well as positive and negative chloride shifts. Okay. Now let us first understand what exactly we mean by this. We have read the paragraph which is given in NCERT. Oxygen dissociation curve is actually the expression or representation of dissociation of oxyhemoglobin, and we know oxyhemoglobin is a temporary complex. So how that complex dissociates, that we need to understand, and when this complex dissociates, what are those conditions and what kind of a graph are we going to get. So let us first talk about this representation which we talked of. This we discussed when we were talking about exchange of gases as well as when we were talking about transport of gases. This is the alveolus. Okay. Now here it is the fresh air which comes in. Now the air which is here has PO2 104, that is high PO2, and PCO2 is 40, low PCO2. Okay, oxygen diffuses in, oxygen comes here, it binds with hemoglobin, and what is formed is oxyhemoglobin, and this oxyhemoglobin gets transported, and here it dissociates to release oxygen and hemoglobin. So oxyhemoglobin is dissociating near the tissue. Is this part clear? Is this much part clear? We'll do it in bits and pieces so that we understand everything. Only this much part, that oxygen diffuses, it enters into the blood, majority of it is transported as oxyhemoglobin. We have seen that 97 to 99%. So oxyhemoglobin is formed, this oxyhemoglobin is transported through blood, right, and then it comes into the tissue. Saturation means how much of hemoglobin is attached to oxygen. Suppose there are 100 molecules and 90% are attached to hemoglobin, so we call it 90% saturation. If all of them are attached to oxygen, then we will call it 100% saturation. Okay, now what happened here at this place? Carbon dioxide from the tissue diffused into the blood, because here PCO2 is high, 45. So now carbon dioxide comes here, right? Now at this place, what happened? At this place, this is the RBC. This carbon dioxide moved into the RBC, CO2 gone inside RBC. It reacts with water which is present in RBC to form carbonic acid. This is happening inside RBC, okay, here at this particular place. That is when carbon dioxide from the tissue is diffusing into the blood and oxygen from the blood is going into the tissue. This H2CO3, that is carbonic acid, dissociates into two parts, H plus and HCO3, that is bicarbonate. These bicarbonate are going to come out of RBC. If they come out of RBC, that means where are they going? They are coming in plasma. So HCO3, they come in plasma. Now if many bicarbonate ions are in the plasma, don't you think the ionic balance of plasma will get affected? It will. So how do we manage that? So if there are 1000 bicarbonate ions coming in, to balance those out, equal number of chloride ions move in, equal number. From where? From plasma into the RBC. Bicarbonate ions are formed inside the RBC, they come out in the plasma. To balance out the number, equal number of chloride ions will diffuse into the RBC. So chloride from plasma move into RBC. This is called chloride shift, and it was explained by Hamburger, so we call it Hamburger shift. Okay, let us not put plus or minus here, positive or negative here, we will see. Now, in this carbon dioxide is being transported, and here oxygen was transported. So you call this blood as oxygenated blood, and this which is going back, you're calling it deoxygenated blood. Okay, now this comes here. Now what do you want to happen at lungs? You want carbon dioxide to be exhaled. Where is carbon dioxide? Right now all that is in the form of bicarbonate ions in the plasma. So let us see what happens here. Again, let us see this RBC at this position, that means closer to the lungs. What is in the plasma? This bicarbonate ions. There are H plus ions because this H also comes out, some number. So H is also out. So now this diffuses in, this diffuses in. Okay, this H comes from one more place, and that is oxyhemoglobin. It acts as a mild acid. It releases H plus. So H plus are coming from oxyhemoglobin also. So H plus go in, bicarbonate also go in, H2CO3 carbonic acid is formed. This carbonic acid will dissociate into two things, water and carbon dioxide. Okay, and these two things, water and carbon dioxide, are exhaled. So when we breathe out, our breath contains carbon dioxide and water vapor also. Is it having water vapor? And that is what you use to clean your specs. Sometimes there are some finger marks, so what you do is you exhale on this glass and then you clean it. So when you exhale, you see those tiny droplets there? Those are the water vapors which have condensed. So our breath also contains water, it also contains carbon dioxide. Now chloride ions are going in. When chloride ions came out from RBC to plasma, to maintain the number of ions, chloride ions were sent in. Now if bicarbonate ions are moving into the RBC, that means in the plasma number of negatively charged ions is going to decrease. So equal number of chloride ions should come out. So now chloride ions are coming out from RBC to plasma. Is it not reverse of this one? So here we start putting a plus sign. This is called positive chloride shift. This is called negative chloride shift. Is this part clear? If yes, then we come to the conditions. Is this part clear to everyone? Then we'll come to the condition and then we will talk about the dissociation curve. Yes, Vidhi, good afternoon. Okay, now let us talk about this condition. Here, oxyhemoglobin is dissociating. Okay? Dissociation of oxyhemoglobin. What are the conditions here at the tissue level? Tell me, what is PO2? High or low? We don't have to worry about the number. How is PCO2? How is the temperature here? And what is the H ion concentration? Tell me. Anju says understood. Very good. So what is the PO2? High or low? At tissue level. Dissociation is taking place at tissue. How is PO2 at the tissue? High or low? Numbers are not required. What is the partial pressure of oxygen? Hiren says low. Very good. So low PO2. What is PCO2 like, high or low, and it is comparative, right? What is the PO2, PCO2 in blood and at this level? How is the partial pressure of carbon dioxide? High, very good. How is the temperature? Higher or lower as compared to the lungs? Okay, Vidhi also says higher. So temperature is also higher, and this H is also coming out. So H ion concentration should also be more. These are the conditions when oxygen dissociates. Okay? Now let us talk about the conditions when oxyhemoglobin is formed. That is here. Formation of oxyhemoglobin. Where is this taking place? At the lung level. Now tell me the same 4 points: PO2, PCO2, temperature and H ion concentration. What is like the partial pressure of oxygen? Is it high? Yes. What is partial pressure of carbon dioxide? Lower. How is the temperature? You just breathing in the fresh air, and at the tissue there are activities taking place which result into release of energy in the form of heat, and that is why the temperature here was higher. So here temperature is comparatively lower. What about the H ions? So these H ions which are released by oxyhemoglobin, they have gone inside RBC. So H ion concentration will also decrease. Okay? So are the conditions clear now? Clear? Very good. Now let us take some numbers. And those numbers are for how much oxygen is still there with hemoglobin. At this point, when we are talking about oxygenated blood, we know PO2 is 95. Remember this? Here, at the tissue level, PO2 was 40. Right? This is what we have talked of. So, when PO2 is 40, here also PO2 was about 40. So, when PO2 is 40, you calling this blood deoxygenated. When PO2 is 95, you calling it oxygenated. Clear? Now let us see. If PO2 is 40, how much hemoglobin is still oxygenated? Oxygenated means it is still attached to oxygen. Let us come back to this. If PO2 is 40, where is this PO2 40? In deoxygenated blood. Deoxygenated blood has PO2 40. So when we say or when we use the term deoxygenated blood, it sounds like the blood has no oxygen. It still has this much saturation. That means 75% of hemoglobin is still attached with hemoglobin, is saturated. And saturated with obviously hemoglobin, and we talked about this. This was called oxygenated blood. So when it is oxygenated, then it is 100% saturated. That means all hemoglobin molecules are attached to 4 oxygen molecules. But when it is like this, that means only 25% has diffused into the tissue. 75% is still with the hemoglobin. Clear? This is what is oxygen dissociation curve. And the questions which are asked are, what are those factors which will bend this curve towards right? That means what will happen what will happen that the curve bends towards right? That means it immediately turns towards right. Or in other words, what are the conditions when oxyhemoglobin will dissociate faster? Can you tell me? What are those things? What are those conditions when the oxygen dissociation curve moves or bends towards right quickly? Instead of normal, it goes like this. Can you tell me the conditions? It is not pressure, it is partial pressure. Okay? It is not pressure, it is partial pressure. Now, if oxyhemoglobin is dissociating, that means the curve immediately bends. That means immediately dissociation takes place. What can be the conditions when oxyhemoglobin dissociates faster? If oxyhemoglobin dissociates faster, instead of a normal curve, your curve is going to bend towards right quickly. Low PO2, high PCO2, high temperature and high H ion concentration. Check your answers. Reshma has written PO2 high, not possible. If PO2 is high, hemoglobin binds with oxygen. We are talking about dissociation of oxygen. See, oxygen dissociation curve basically means dissociation of oxyhemoglobin. High H plus, high temperature, high PCO2, low O2. So these conditions will result in turning of the curve towards right. Clear? Is oxygen dissociation curve clear? Is positive negative chloride shift clear? So when you talk about positive chloride shift, bicarbonate ions move from RBC to plasma and chloride ions move from plasma to RBC. In negative chloride shift, bicarbonate from plasma into RBC and chloride ions from RBC to plasma. And now when you read this, okay, so now let us read this. This clearly indicates that oxygen gets bound to hemoglobin at the lung surface and gets dissociated at the tissue. Every 100 milliliter of oxygenated blood can deliver around 5 milliliter of O2 at the tissue level under normal conditions. The remaining oxygen remains with hemoglobin. This number is very, very important. Only 5 milliliter of oxygen is released at the tissue level under normal conditions. This value is going to change if the physiological conditions change. Now this paragraph you need to read again after the class so that if anything is not clear, that also will be clear, and in case if there is any doubt, then we can take that up tomorrow's class. Okay, so if this part is clear, then we will start with Bohr's effect and Haldane effect. Okay, so let me know. Hiren says it is clear. Amisha says it is clear. What about others? Everyone has understood this? Clear? Reena also says, Anju, Amisha, Hiren. 4 of you have said that it is clear. I don't know about others. You need to just message so that I know it is absolutely clear. Reshma also says it is clear. Is it the same, Mr. Bohr? Yes. The scientists were like, they were in genius categories. So they did things in biology also, they did things, they found out, discovered things in chemistry also. So they are like the same ones, but yes, the contribution is different. Okay, so now let us talk about Bohr's effect. Oxygen binding capacity of hemoglobin is lowered or decreases one and the same thing. You write lowered or you write decreases with increase in PCO2, that is with increase in partial pressure of a carbon dioxide, oxygen binding capacity of hemoglobin is going to decrease. Now, is this statement telling us anything else? Where is this happening? Where is this happening? At the tissue level. Carbon dioxide is being released in the tissue, and the blood which is coming at the tissue has oxyhemoglobin. So as partial pressure of carbon dioxide increases, that oxyhemoglobin is going to dissociate, because hemoglobin's binding capacity or oxygen's binding capacity of hemoglobin decreases as soon as carbon dioxide partial pressure increases. So this is known as Bohr's effect. It is seen at tissue level. That means if a tissue is more active, PCO2 is going to be even higher, PO2 is going to be even lower. That means more and more oxyhemoglobin will dissociate. So depending upon the requirement of the tissue, oxygen will be released. If it doesn't require that much, PO2 is 40, so little bit 5 milliliter of oxygen will be released. If the tissue is very active, then it requires more oxygen. So PCO2 will be more and more oxyhemoglobin will dissociate, more and more oxygen will be released. PO2 will also be less. So if PO2 is 40, we know what is the percent saturation. If PO2 is 20, we know what is the percent saturation. So Bohr was the one who showed or who explained that oxygen's binding capacity of hemoglobin decreases when partial pressure of carbon dioxide is higher, or when partial pressure of carbon dioxide increases. Okay, And when PO2 is high, oxygen dissociation curve is going to bend towards right. Alright, let us come to the other one, that is called Haldane's effect. Is Bohr's effect clear? It actually tells us the reason for release of carbon dioxide. And where is carbon dioxide released? At the lungs. So Haldane showed or said that oxyhemoglobin acts as an acid. Okay? As an acid, it releases H+. This H+ and HCO3, they move into RBC. Here they react to form H2CO3, carbonic acid, which dissociates into CO2 and water, and these two are exhaled. So are they not talking about reverse of chloride shift, or is he not talking about negative chloride shift? So the process was this. Haldane did not give any reference to chloride. He just said that oxyhemoglobin acts as an acid, releases H+, H+ diffuse into RBC, bicarbonate diffuse into RBC, and carbon dioxide is released, and water also. So this is Haldane's effect. So Bohr's effect and Haldane's effect. Are these two things clear? Okay, Hiren says super clear, and he has sent me some glowing bulbs also. It's like dimag ki batti jal gayi types, okay? It is very important, very, very important, Amisha. Alright, if that part is clear, then let us come to control of respiration. Clear, 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 very good. Clearly understood all concepts, perfect. Let us now talk about how our respiration is controlled. There are two control mechanisms. One is nervous, other is chemical. So we are first talking about the nervous. And when we talk of nervous, that means we are talking about the nervous system. And if we are talking about nervous system, that means there are some centers in our nervous system which are responsible for controlling our respiration, our breathing. Because when we inhale, the muscles have to contract, and for muscles to contract, the message has to come from the brain. So if you are talking about our nervous control, then there are three centers in our nervous system. The first center is known as inspiratory or inspiratory center. The second one is called expiratory or expiratory center, and the third one is called pneumotaxic center. So these are the three centers which are on in our brain, and they are the ones which are actually going to help in the respiration, that is the nervous control. The first and the second, they are located on medulla oblongata. This one is also located on medulla oblongata. Okay, MO is the abbreviation that we use for it. And pneumotaxic center is located on pons. That is pons varolii. Very good. Anju has answered that it is located on pons. All right. Inspiratory center stimulates contraction of the inspiratory muscles. And which are these inspiratory muscles? I don't know why sometimes my brain shifts from automatic breathing to manual breathing. What does that mean? What do you mean by automatic and what do you mean by manual breathing? Hiren? How come your brain shifts from automatic to manual? Yes, phrenic and external intercostal. Perfect. So the stimulus is going to come from inspiratory center. It is located on medulla. And this center is normally responsible for maintaining normal rhythm of our respiration. Maintains normal breathing rhythm. So inhalation, exhalation, when we are doing other activities, when we are listening to something, when we are writing, when we are talking, we are not even thinking about this breathing, still that normal inhalation, exhalation continues. And this is the job of inspiratory center. Okay? Now let us talk about this expiratory one. It normally remains inactive during normal breathing. Remains inactive during normal respiration. Because normal respiration is taken care of by the inspiratory center. So this expiratory center, it remains dormant or inactive during normal breathing. It just gets activated when it is not normal. So what happens when it is not normal? What kind of breathing are we talking about? It gets active during forceful breathing. If you're doing anything forcefully, forceful inhalation, forceful exhalation, then this center gets active. So normal breathing, normal inhalation, normal exhalation, that is contraction of phrenic muscles, external intercostal, their relaxation, everything is controlled by inspiratory center. But if forceful breathing is taking place, then it is expiratory center which gets active. So it is responsible for that forceful exhalation. That means which muscles are contracting? So the message for contraction of internal intercostal muscles and abdominal muscles, their contraction is controlled by expiratory center. Okay, now this pneumotaxic center, it results into panting. Panting is very fast breathing. it causes panting, fast breathing. You know, I'm sure you've seen dogs, they breathe during summers, they breathe very fast. They put their tongue out and they continuously, very fast, they breathe. And they do it because from that tongue saliva evaporates, and evaporation takes the heat from their body, so it gives them a cooling effect. That is called panting. So if you do some exercise, if you are jogging, if you're running, and after that when you stop, you feel that you're breathing at a very faster pace. So that is called panting. Rate of respiration increases very, very fast, but duration decreases faster, so shorter inspiration, shorter exhalation. Rate of respiration increases but the duration decreases. This is called panting, and for this the center is located in our pons. So our nervous system controls breathing. All right? Is this part clear? The normal process is regulated by inspiratory center. The expiratory center is responsible for forceful breathing, especially forceful exhalation, and pneumotaxic center is for panting. Location: 2 are located on medulla and 1 is located on pons. Okay, now let us take the other control. It is called chemical control. So our breathing also has a chemical control, and okay, Reshma is saying मेरा keyboard खराब हो गया था. Okay, so you wanted to ask some questions which you could not type. Okay, Vidhi, it's clear to you. That's very good. So Reshma, you have some questions which you wanted to ask. Okay, it's all clear. Now, the chemosensitive areas that means the areas which are very sensitive to chemicals are located in respiratory rhythm centers. Okay, respiratory rhythm centers, and this is like they are very, very sensitive to carbon dioxide concentration of carbon dioxide. Now, where are these chemosensitive areas located? At the origin of aorta and carotid artery, and that is why these centers are called aortic body and carotid body. Now, if you remember the structure of human heart, this is the place from where the aorta rises, right? And then it bends towards the left side, and if you remember here, there are 3 main arteries. Okay, this is aorta and this is carotid. So now at the base of it, at the base of these, here, there are these bodies. These are specialized cells and they are very sensitive to concentration of carbon dioxide, not to oxygen. So our chemosensitive areas are sensitive to carbon dioxide, though the main respiratory gas that we need is oxygen. So if the blood which comes here is not properly oxygenated, this is the compartment which is going to receive oxygenated blood from lungs through pulmonary veins. So if here there is blood which is having higher concentration, blood with higher concentration of carbon dioxide, then the place where this blood enters is aorta, and right here is that body which is going to detect it. Then this blood comes here and this carotid is going to take it to our head. Through the neck region, it is going to come upwards. So it has to check whether proper oxygenated blood is going to the brain or not. So if this detects that the blood has more carbon dioxide, then the respiratory rhythm centers will be stimulated. Okay? And how does it work? Suppose there is more of carbon dioxide. Suppose carbon dioxide increases. Then we know carbon dioxide is going to react with water to form H2CO3. Less of this H2CO3 will be produced because the enzyme carbonic anhydrase is mainly present in RBC. But as soon as H2CO3 is produced, it dissociates. And if in the blood H ion concentration increases, that means the blood becomes more acidic. And this is known as acidosis. It is called acidosis. What is happening is the pH of blood decreases or becomes acidic. Becomes acidic. Okay? Hiren is saying there is high concentration of oxygen in left ventricle. Yes, because the left auricle receives oxygenated blood and that blood comes into the left ventricle. Okay, and whenever this condition takes place, the respiratory centers are stimulated and rate of respiration increases. And this rate of respiration increases to remove excess of carbon dioxide. Okay. Oh, here, I purposefully wrote CO2. Left one, Hiren, there is a confusion here. We are talking only about the chemosensitive areas. Normally, through the lungs, oxygenated blood enters into the left auricle. From the left auricle, it will come here. But if the blood is with higher carbon dioxide, then these areas will be stimulated. That is why I wrote CO2. Okay, in normal case it should be oxygen, but here because it is not a normal condition, and if the blood has more of carbon dioxide, then only they are sensitive to carbon dioxide, then only they will be stimulated. So now our body knows that the blood has more than required carbon dioxide, so that excess of carbon dioxide has to be exhaled, and that is why the rate of respiration increases, and that is to remove that excess of carbon dioxide. Yes, we were talking about this situation when they are sensitive to carbon dioxide. Clear? These centers, the chemosensitive areas or our respiratory rhythm centers, are either insensitive or least sensitive to oxygen. That means our breathing rate is going to be normal if the blood here has normal oxygen, normal carbon dioxide. Our breathing rate will increase if the blood entering into aorta has more of carbon dioxide. This is what is regulation. Our breathing has to be regulated. If everything is normal, the inspiratory center is taking care of it. But if the conditions are different, then there are various things which are going to control our rate of respiration. I'm sure you know what is yawning, and why would somebody yawn? Can you give me a reason? What is yawning and why do we yawn? And what is that time when we should be yawning? It's a body's mechanism, so you do not decide, it is the body which decides. So biologically, what is the normal time when you should be yawning? Reena says less O2. So if there is less oxygen, then you should be yawning, right? I'm not saying this is right. I'm just taking the things. Hiren is saying morning, okay? I thought you would say during the class, because students start yawning in the class. After waking up, when you feel lazy. Yawning actually helps in removal of excess of carbon dioxide. So whenever in your body, in your blood, in your tissues, there is more carbon dioxide, then you tend to yawn. And yawning helps in removal of carbon dioxide. Now tell me, when would be carbon dioxide more in your tissues? When the tissues are working. You wake up in the morning, you were lying down, your muscles were relaxed. So why do you think there is more carbon dioxide produced? Your muscles are relaxed. If muscles are relaxed, they would use less oxygen, they would give out less carbon dioxide. It is when your muscles are working continuously, that is when they keep taking oxygen and keep releasing that carbon dioxide, and that carbon dioxide keeps accumulating in our body. So biologically, the correct time of yawning should be late evening, when you have done your entire day work, you were continuously sitting, because when we are sitting, our back muscles are all the time contracted. You're talking, you're writing, that is when your muscles are working. So biologically, when the muscles have worked for a long period of time, there would be accumulation of carbon dioxide, and when this carbon dioxide has to be released, that is the time you should be yawning. So late evening when you're like tired and you feel like sleeping, that is the time you should be yawning, not in the morning. So this is the way you need to know your body's physiology. This is biology. This is what we are studying. So some people, they wake up in the morning and they're continuously yawning. That means if you wake up and you're still yawning, that means you did not get a good night's sleep, your muscles did not relax properly. After having a good night's sleep, the muscles should be relaxed and you should not be yawning. Okay? So there are two mechanisms by which our respiration or rate of respiration is controlled. One is nervous, other is chemical. And our chemosensitive areas are sensitive to carbon dioxide, not to oxygen. And remember this term acidosis? It is when the pH of blood decreases due to release of more H ions. And this happens when carbon dioxide accumulates in the body. Okay. Now in this complete part that we have talked of today, is there any confusion? Anything which is not clear? Okay, Anusha is saying everything is clear, no problem. Reena also says everything clear, that's very good. What about others? Hiren also says it is clear. Reshma, no doubts. But don't forget, you have to do one thing today after the class or whenever you get time today, that you need to go over this entire process through the notes as well as read the NCERT and read it carefully so that each and every sentence which is written is absolutely clear to us. Now in this particular chapter we are left with only one thing, and that is respiratory disorders or disorders associated with our respiratory system. Now what kind of disorders can we talk of or can we think of? Diseases like pneumonia, tuberculosis, anything else that comes to your mind which are respiratory system related disorders. Have you heard of asthma? Yes, asthma, allergy. Have you heard of occupational disorders? Occupational disorders. Yes, asthma, bronchitis, pneumonia, tuberculosis, these are all respiratory system related disorders. But there is something called occupational disorders. You know what occupation is? Occupation is the job that you do. So sometimes some people, they are into a job where they get affected or their respiratory system gets affected. For example, if some people are working in, say, a factory where silicon is released or where asbestos is released or where cotton dust is released, then when that fine dust or when that powder enters into our respiratory tract, then that disorder is known as silicosis, asbestosis, depending upon the particle, and only those people will be affected who are working in that factory. If you and I, we are not working in that factory, we are not living in that area, we will not get affected. So it is the job or the occupation which is responsible for it. So such are called occupational disorders. Then there are people who smoke. So smokers also have a condition associated with smoking, and that is called emphysema. So all these things we will talk about when we come to disorders. Yes, coal mines due to dust normally results into allergies. Yes, lung damage. So coal mines, cotton mills, cotton ginning mills, the place where asbestos sheets are made, or many such areas where a very fine powder is released in that air, and people working in that area, they continuously inhale that thing. So those will be occupational disorders. So what we will do tomorrow is we will start with the disorders related to our respiratory system, all the diseases, all conditions, and then maybe I'll give you a short quiz on this chapter. So the disorders is first thing that we will talk of, then we will take doubts in case if you come across anything, and then I'll be giving you the quiz. Hiren is saying, but Delhi everyone is facing without having such. See, there are certain things which are taking place. The states which are around Delhi, that is Punjab and Haryana, these are states where agriculture is the predominant thing. And I'm sure you've seen that after a crop is harvested, those parts which remain of the plant, they are cut and they're burned in the forest or in the field itself. So when you're burning something, it results in smoke, and that smoke has unburnt carbon pieces. So it is again a condition. This is not a normal thing, like in hectares and hectares of land, if you keep burning all those plants, then it is going to result into that complication, right? But normally, if you don't do this, then such kind of things would be seen only if a person is into that occupation. Otherwise, those problems will not be seen. Okay? So that is the problem with people who are living in Delhi and NCR are facing right now. Okay, so go over all these things in your NCERT, and when we meet tomorrow, we will start with the doubts if you have any. Respiratory disorders we will take up next. And if you have not decided for the subscription yet, you need to do it fast. And this is my name, my code, which you can use while you take the subscription so that you get the 10% discount. Alright, so I will see you tomorrow, and we will finish this chapter and start preparing, start revising so that you are able to answer the questions on the quiz. Okay, so see you tomorrow. Bye.